1, so we're back at it, and I feel a bit more energized, so I feel much better right now than like an hour ago or 2 hours ago. So let's continue with the immune system. So we had here that there are 5 types of antibodies. We have the IgA, IgE, IgD, IgM, and IgG, right? So G is just with the G itself, and A, we start with A. So we have those 2 down, and D and E also rhyme with G, so we want those as well. And M is an outlier, so just know there's IgM too, right? For now, that should do the job. Now, IgM is the first antibody made against an infecting pathogen. So we have the IgM, which is the first antibody made against an infecting pathogen. That's interesting. So IgM is the first thing that you make. Then we have somatic hypermutation, which selects for antibodies that bind more tightly to the pathogen. So if you have antibodies that bind more tightly to the pathogen, you have somatic hypermutation. As you can see the difference here, the cleft is more is wrapping around the antigen more than IgM, which was the first antibody. And then switching antibody isotype to IgG allows delivery of the pathogen to phagocytes. So switching the isotype of the antibody to IgG, it allows for, so we had a variable region, right? The variable region here you can see is changed again, and there is a heavy chain attached to the IgG receptor, and this allows for delivery of the IgG to of the pathogen to phagocytes. But for now, we need to know that. But the main concept for this flashcard was that hypermutation is the mutation for the antigen binding site, the variable region on an antibody. So what does ingest pathogens? Break them down into antigens, then present those pathogen specific antigens on MHC2. So we know that it is macrophages that ingest pathogens, and they break them down into antigens and present them as MHC2. So yeah, for now we have this down. So what I had in my head instead of macrophages was kind of antibodies, or I don't know, like some natural killer cell, or maybe something else. I forgot. Instead of macrophages, but like phagocytes, like stuff like that. We know macrophages do phagocytosis. So yeah, it's important to know that macrophages represent the MHC2, and they break down the pathogens. The main function of Watt cells is to process antigen material and present it on the cell surface to the T cells of the immune system. I still have the picture in my head. It's the dendritic cells. So the main function of dendritic cells is to process the antigen and present it to the T cells, and they act as the messengers between the adaptive and the innate immune system. So what are a type of lymphocyte? They can recognize and kill cancer cells or other cells that have been infected. And we know that this type of lymphocyte is a natural killer cell that recognizes and kills cancer cells. So the thing about cancer cells is that when our cells are cancerous, or they have been infected by a virus, natural killer cells do not attack invading organisms directly, but instead they destroy the body's own cells that have either become cancerous or been infected with a virus. So they destroyed the body's own cells, own cancer cells, or when they are infected by a virus. So the killing of our own cells is called apoptosis. And in order to make sure we have this down, yeah, I forgot. Oh, here it is. So we can write what is apoptosis. Is it the killing of our own body cells that are cancerous or have been infected by viruses? And these cells kill when there is no MH. So MC1 here, I'm assuming it's MHC1. So they kill when there is no MC1 and when a cell is giving off stress proteins. So ligands, as we saw here, both of them have to occur because no MHC and activating ligands, then you attack the target. But if there's normal MHC1 and then activating ligands, there's a dynamic equilibrium. That's why the creator of this flashcard was also confused. So we can ask, yeah, we asked it there, so that should be enough to see what that is about. So for now, we're good. What is a blood protein produced in response to a specific antigen? So we know because it's in the blood and it's a protein and is specific to an antigen, it is antibody. So we're talking about antibodies here. And we know that they have variable regions. They have a light chain and a heavy chain. The heavy chain is like what is stabilizing the antibody, and the light chains are on the two sides as well. And then the variable region is the section at the end where it attaches to wherever it needs to attach to. Yeah, we can come back to that later. For now, we're good. What is a term that refers to an immune process where pathogens are targeted for destruction by an immune cell known as a phagocyte? So the process that we're talking about here is tagging the pathogens for destruction by an immune cell known as a phagocyte. So opsonization is that process. Okay, now we know what opsonization is as well. So antibodies mark pathogens for destruction, and opsonization is A. B is adhesion and ingestion. C is phagosome formation, and D is phagosylysosome formation. And we can see here that the IG immunoglobulin G, IgG, is the red Y-shaped antibodies here. Then we have the FCY receptor for the adhesion and ingestion stage. We have the complement proteins on the antigen on the pathogen. We have other opsonins, however you say those, pronounce those. Then we have also the complement receptor, and the enzyme pool is here where the lysosome, the phagolysosome, phagosylysosome is formed. And yeah, so we have IgG and IgM at the end, and the antibodies mark pathogens for destruction. What is when pathogens clump together into insoluble complexes? So when pathogens clump together into insoluble complexes, it is called the probably opsonin agglutination. My bad. It's caused by opsonizing pathogens. So we know that opsonization was so we had opsonization, and we also have the agglutination. So agglutination is when pathogens clump together into insoluble complexes. So they activate or direct agglutination. So they're caused by opsonizing pathogens. Now, I immediately forgot what opsonization was about, but the tagging is opsonization. Tagging the pathogen in order to be phagocytosed is opsonization. But agglutination is the pathogens clumping together into insoluble complexes, and this is caused by opsonizing pathogens. So the pathogens that are targeted, that are tagged for opsonization, they cause the agglutination. So this is the active or direct agglutination, and as you can see, it is the antibodies that help with this agglutination, that do the agglutination, basically. What are a B cell subtype that are formed following primary infection and are important in generating an accelerated and more robust antibody-mediated immune response in the case of reinfection? So what is a B cell subtype that are formed following primary infection? So excuse my coughing. I know that the COVID still is in my body. So a B cell subtype that is formed following primary infection and is important in generating an accelerated and more robust antibody immune response In the case of reinfection, it should be the memory B cells. Yes. So they are formed following primary infection. So the first time that you are infected, they are formed. Memory B cells are formed, and memory B cells are important in generating an accelerated and more robust antibody-mediated immune response in the case of reinfection. So your antibody mediated immune response takes place during the first primary infection, but it is even more robust and accelerated when the memory B cells work in case of reinfections. So there's a pixelize for this apparently. So we have memories and memory B cells as memories, which stores old information, old photographs. It's also long-lived. It shows the old age. I'm assuming that memory B cells are long-lived. And we have primary immune response, so bits of virus or bacteria. We have naive B cell, we have naive T cell, and then when we have memory T cell and memory B cell, you can see the antibody being attached to it. You have second exposure to bits of virus or bacteria, and this secondary immune response has lots of B cells and T cells. So we're good for now, and we got it, so we can do this later. We have what immunity that produces an immune response that does not involve antibodies, so it should be innate immunity. So we have cell-mediated cytotoxic immunity that produces immune responses that do not involve antibodies. This involves the activation of phagocytes, T cells, and the release of cytokines in response to an antigen. And as you can see here, this is the pathway. So it involves the activation of phagocytes, T cells, and release of cytokines. So your phagocytes need to be activated. This happens with the cell-mediated immunity. Your T cells need to be also activated. This is also with cell-mediated immunity, cytotoxic immunity. And your cytokines need to be released in response to an antigen, and that also happens with cell-mediated immunity. So you have here the first step: a T cell binds to MHC antigen complex on the surface of the infected cell, activating the T cell with a cytokine receptor. We have helper T cell produces cytokines that cause the activated T cell to differentiate into a cytotoxic T cell. These cytokines also influence the formation of plasma cells and activated macrophages. As we know, plasma cells were responsible for making antibodies. They are mature B cells. We didn't get this, so we can repeat that. What are a type of white blood cell that circulates around our bodies, scanning for cellular abnormalities and infections? This one, I'm not sure. It might be the mast cells. No, it's T cells. So we have examples of T cells. These are the 4 examples, 4 subtypes that we need to know of. There is the helper T cell. We know that helper T cells help the B cells to make antibodies, right? We have cytotoxic T cells. We have suppressor T cells. And we have memory T cells. So they circulate in our body, right, around our body, and they scan for cellular abnormalities and infections. So they're just hovering around in our body, trying to find abnormalities. Now, we have microbes that are ingested by macrophages. These macrophages have self-proteins, and the antigen from the microbe is a non-self molecule. It is recognized and identified. And then the second step is that these antigens from microbes are taken by antigen presenting cells. They form a self-non-self complex, and they go on the surface of the macrophage, and this macrophage is now turned into an antigen presenting cell because it's representing an antigen. Then there is interleukin 1, which stimulates helper T cells, and helper T cells have T cell receptors, and they bind. They have binding site for antigen, and they have a binding site for self-protein. And interleukin 2 stimulates cell division, and interleukin 2 also activates other B cells and T cells. So the B cells give humoral immunity, the secretion of antibodies by plasma cells, and the cytotoxic T cells give cell-mediated immunity, and they attack on infected cells. So helper T cells are the main T cell that give rise to both B cell and cytotoxic T cell. Now, before the helper T cell being on its own, it has a receptor that attaches to the antigen presenting cell that was once a macrophage, and the T cell receptor attaches to the self-non-self complex, which this is where exactly the helper T cell is able to differentiate into cytotoxic T cell and B cell, because it has already interacted with the antigen. And this is very interesting, to say the least. Okay, we have this to do again. We have positive and negative selection of T cells. So what is a positive selection? What is a negative selection? A positive selection is when only mature T cells, so mature only T cells that can respond to the presentation of an antigen on MHC. So you mature only T cells that respond to presentation of antigen on MHC. But negative selection causes apoptosis in T cells that are self-reactive. Interesting. So T cells have this thing called positive selection and negative selection. We have maturation of T cells. So when you have maturation of only T cells that present the antigen on MHC, then you have positive selection. But negative selection is when apoptosis happens. So negative selection causes apoptosis in T cells that are self-reactive. So some of these T cells are self-reactive. So they react to the self cells in our body, and that is why they are called negative selection, because they die. Apoptosis happens for them. So they are facilitated by thymosin, thymosin, and it occurs in thymus. And thymus is, I believe, somewhere near our thyroid, right? So yeah, the maturation of T cells is facilitated by thymosin and occurs in thymus. Whereas thymus and how does thymosin mature T cells are T cells produced in our thymus or elsewhere. So we have immature thymocyte, then there is a T cell receptor, CD8 and CD4, that comes on top of the immature thymocyte. Then there's positive selection of cells whose receptor binds self HLA molecules, and there is apoptosis of cells that do not interact with HLA molecules. So what are HLA molecules? During positive and these HLA molecules, there is negative selection in apoptosis cells with high affinity receptor for self HLA and self antigen. So the mature CD4 plus or CD8 plus T cells are formed on the left side, but the negative selection in apoptosis does not form any mature T cells, neither CD4 plus or CD8 plus. You can watch the Khan Academy on this later. So what activates cytotoxic T cells? So what activates cytotoxic T cells and macrophages to attack infected cells, or they stimulate B cells to secrete antibodies? So the activation of macrophages in cytotoxic T cells to attack infected cells. I'm assuming it is helper T cells. Yes, they are the ones that activate, if you remember the diagram that I just explained, where the helper T cell gives rise to both B cell and cytotoxic T cell, which was also itself connected with the antigen representing macrophage cell. So you also write helper T cells TH or CD4 plus. So now we know that CD4 plus is used to describe helper T cells. So Again, helper T cells activate cytotoxic T cells and macrophages to attack infected cells, or they stimulate B cells to secrete antibodies. So B cells stimulate helper T cells stimulate B cells. They do not give rise to them actually, but they are affiliated with B cells, which help them to secrete antibodies. But also they activate T cells. They also do not give rise to T cells, cytotoxic T cells, but they activate them. So they activate macrophages or cytotoxic T cells, and they are able then to attack infected cells. We got this, so we're going to put this. What is the term used to describe mutation of the antigen binding site? The variable region on an antibody is called hypermutation. We know it now. And the first antibody that is made against an infecting pathogen is IgM, and then IgG is the most stable, right? It allows for delivery of pathogen to phagocytes. So they allow delivery to phagocytes. What responds to antigen on MHC1 and kill virally infected cells? So CD8 plus should respond to cytotoxic T cells, killer cells. TC is another way to write it. CTL, cytotoxic lymphocytes, T cells, I believe, or CD8 plus. They respond to antigen on MHC1 and they kill virally infected cells. So any cell in our body that is virally infected, T cells, cytotoxic T cells, TCL, or CD8 plus, they just come and kill it. They're like the Navy SEAL team that killed Osama bin Laden. I don't know why I said that, but just this is me saying that. Osama, you're a virally infected cell, and we're gonna kill you because we are CD8 plus. Cytotoxic T cell binds to infected cell. So we have the cytotoxic T cell that binds to the infected cell, and then perforin makes holes in infected cell's membrane, and then infected cell lyses. So this is the mechanism of the cytotoxic T cell. It binds to the infected cell. The perforin makes holes, permeates the cell membrane of the infected cell, and the third step, which is the final kill shot, it is the infected cell lysing, right? So what does downregulate the immune response after an infection and promote self-tolerance? It should be the memory B cell, suppressor T cell, the regulator T cell. It downregulates the immune response after an infection and promotes self-tolerance. So they help to, you also write them as Treg, like T-Rex, but they help to distinguish between self and non-self molecules, and by doing so reduce the risk of autoimmune diseases. So if you're asked what could possibly reduce the risk of MS or Hashimoto's disease, two good examples of an autoimmune disease, then you know that it is the regulator T cells. They reduce the risk of autoimmune disease. How do they do it? By helping they help to distinguish between self and non-self molecules. And that also downregulates the immune response after an infection and promotes self-tolerance. They are suppressor T cells. So regulatory T cells. Treg has anti-inflammatory function. The regulatory T cells maintain homeostasis of immune response. So we have inflammatory response. They have anti-inflammatory function. Nice. That's why you don't want any autoimmune disease activations, right? Cool. So we're going to... So what serves a similar function to what, and a second encounter with a pathogen will work to mount a faster and stronger immune response than the first time? It should be antibodies. Memory T cells serve a similar function to memory B cells. And at a second encounter with a pathogen, so it's not only the second encounter, but at the subsequent encounters with the pathogen, they will work to give a faster and stronger immune response. And the term antigen experienced T cell is also used. So you have memory B cells for sure, but for memory T cells, you can also call them antigen experienced T cell. Perfect. So now let's do this. What is a term that refers to an immune process where pathogens are targeted for destruction by an immune cell known as a phagocyte? So what is a term that refers to an immune process where pathogens are targeted for destruction? So if pathogens are targeted for destruction by an immune cell known as the phagocyte, oh, this is about opsonization. So opsonization is like targeting and tagging the pathogens for destruction by phagocytes. We got this now. So in the case of what condition a self-antigen is recognized as foreign and the immune system attacks normal cells? Oh, this is an autoimmune condition. So in case of an autoimmune condition, a self-antigen is recognized as a foreign and the immune system attacks normal cells. This is a good example. So we have when you're sick and infertile, sperm cell is exhausted there, sitting there. This is a strange meme. We have white blood cells that are targeting somewhere else. So I think they should be targeting him, not the surroundings. I don't get yeah, whatever. So immune response against bacteria, antibodies attaching body tissues. So yeah. Okay. What occurs when the immune system uh, overreacts to a harmless substance known as an allergen? So when you have an allergen, it's a harmless substance, but it overreacts to it. It is an allergic reaction, right? So an allergic reaction is what happens when your body overreacts to a substance that is harmless, known as an allergen. The three stages of an IgE mediated allergic reaction. So IgE is apparently involved with allergic reactions. Think of mast cells in IgE. Remember, IgM has many sides. It's uh, it's in the beginning, and IgG is like a gaping hole used for endocytosis at the end. So um, we have the allergic response, the three stages of an IgE mediated allergic reaction. We have the IgE antibodies. We have the mast cell. We have granules. We have allergens. We have histamines. Yeah. What releases cytokines and granules that contain chemical molecules to create an inflammatory cascade? It should be the cytotoxic, like the T cell. It's the mast cells that release cytokines and granules. So we know that mast cells release cytokines and granules that contain chemical molecules to create an inflammatory cascade, important for wound healing and defense against pathogens via the inflammatory response. So when you want to have wound healing and have defense against pathogens via the inflammatory response, you have the mast cells to release cytokines and granules that contain chemical molecules to create inflammatory cascade. The steps are: first, initial allergen exposure. You can see the allergens being exposed to the B cell. Then you have antibody IgE production as the plasma cell, and then you have histamine producing mast cell. So mast cells are specifically related with allergens, allergic reactions. They produce histamines, and then IgE bound mast cell. So the mast cell is primed. You have histamines, and IgE is also bound to the mast cell, and then the allergen, when it is re-exposed, there is allergic reaction. Histamines are released. So we get hypersensitive during the inflammatory response. So it is the mast cells that release cytokines and granules that contain chemical molecules to create an inflammatory cascade. Is it histamines or granules histamines? Are granules histamines that mast cells produce? Or the other way, are histamines granules that mast cells produce? What is when pathogens clump together into insoluble complexes? It is agglutination. Whenever it is glutinized, it's like clumped together. It is caused by opsonizing pathogens. What is released by mast cells? Histamines. and causes blood vessels to dilate, thereby increasing blood flow and cell traffic into an area of infection. Yes, it's histamines. They are released by mast cells and they cause blood vessels to dilate, therefore increasing blood flow and cell trafficking to an area of infection. So yeah, we can. It's released by mast cells, histamines. Yes, they also cause vasodilation. They increase blood flow to and this cell trafficking to an area of infection. You want that, right? I mean, you don't want it, but it is what it is. That's what they do. What type of immunity produces an immune response that does not involve antibodies? That is a cell-mediated cytotoxic immunity. Perfect. What immunization induces what immunity? The activation of B cells that produce antibodies. It is the adaptive immunity. Oh, immunization induces active immunity. So we have the adaptive acquired immunity. So the first step is vaccination, basically immunization, right? And then we have the nanoparticles of the vaccine that are uptaken by the APC, is the activation, and then the stimulation gives rise to cytotoxic T cells, helper T cells, which help the B cells for humoral immunity and production of antibodies. But cytotoxic T cells also give rise for cellular immunity and release of cytokines. So we now here know that cytokines are also released by cytotoxic T cells. So that's something to keep in mind as well. But immunization induces active immunity. What transports lymph, a fluid containing infection-fighting white blood cells? Infection-fighting white blood cells throughout the body. It should be the lymphatic system. The lymphatic system transports lymph. It is a fluid that has infection-fighting white blood cells, and it transports these white blood cells throughout the body. It equalizes fluid distribution. So you want the white blood cells to go everywhere in our body. That's why you have the lymphatic system. And it transports fats and fat-soluble compounds in chylomicrons. So it also transports fat-soluble components in chylomicrons. You have the tonsils, you have the thymus. Oh, this is where the thymus is. You have lymph vessels, you have lymph nodes, you have the spleen, and you have the bone marrow, which is here. And this is the bone marrow we're talking about, not here, right? You have a venule, you have a lymph capillary, you have the blood capillary, you have the arterial. This might be the axillary. For now, yeah, we notice. What results when the lymphatic system is overwhelmed and can't drain excess fluid from tissues? So when the lymphatic system is overwhelmed and can't drain excess fluid from tissues, this is edema. Interesting. So edema is from when the lymphatic system is puffed and it's overwhelmed and cannot drain the excessive fluid from tissues. So you have the lymph nodes, you have a healthy system, the fluid drains to lymph nodes, and you have a lymphedema where blocked fluid causes swelling. A virus is a non-living particle containing what and what that can infect a living cell. It contains a DNA, we know that, uh, and so a protein and a DNA or RNA. We have HIV, hepatitis B, Ebola virus, bacteriophage, influenza, and adenovirus. So a bacteriophage is also a virus. This I actually didn't know till last month or something. So adenovirus is a virus, influenza is a virus, Ebola, hepatitis B, HIV. These are all viri, and they contain protein and the DNA or RNA. What displays endogenous antigen to cytotoxic CD8 plus T cells? It should be MHC1, and MHC stands for histocomplex, major histocomplex 1, right? Yeah, we got that right. What are a type of white blood cell that circulate around our bodies? Scanning for cellular abnormalities and infections, we know those are the cytotoxic T cells. The T cells, basically T lymphocytes, they circulate around our body. Even now we know through the lymphatic system, and they scan for cellular abnormalities and infections, right? The antigen binding region of an antibody is the variable region. Tip of the variable region is the antigen binding region. So the tip of the variable region. Because we didn't say the tip, we have to do it again. B cells mature into something that produce antibodies. They mature into plasma cells that produce antibodies. B cells mature into plasma cells that produce antibodies. Perfect, we now know that. What stimulates B cells to secrete antibodies? It is the helper T cells that stimulate B cells to secrete antibodies. Perfect. What pathway is a central regulator of the inflammatory response? The arachidonic acid pathway is a central regulator of the inflammatory response. Arachidonic acid is the precursor to eicosanoids. I have no clue what this is about. We know now that what is arachidonic acid pathway. We know that it's a regulatory pathway, a central regulator for inflammatory response. How does it regulate the inflammatory response? And arachidonic acid is precursor to eicosanoids. What are eicosanoids? And what do they do? Okay. Oh, we have positive and negative selection of T cells. So positive selection is selecting any T cells that have the MHC presenting pathogen on them, and negative selection is any of them that don't have it, kind of. So it causes apoptosis in T cells that are self-reactive. But positive selection is mature T cells that can only T cells that respond to the presentation of antigen on MHC. What responds to antigen on MHC1 and kills virally infected cells? So what responds to antigen on MHC1 is the CD8 plus, or this cytotoxic T cell, killer cells, TC, CTL, CD8 plus, cytotoxic T cells. They respond to MHC1. So they attach to the pathogen, cytotoxic T cell binds to the infected cell. It uses perforins to create holes in the cell, in the infected cell's cell membrane, and then the cell lysis, the infected cell lysis, right? What downregulates the immune response after an infection and promotes self-tolerance? It is the regulatory, like suppressor T cells, the regulator T cells, Treg. They distinguish between self and non-self molecules, and that's how they reduce the risk of autoimmune diseases. And what serves a similar function? So memory T cells serve similar function as memory B cells, and at the second encounter of with the infection, they have a stronger and faster immune response than the first time. What releases cytokines and granules that contain chemical molecules to create an inflammatory cascade? It is our T cells, mast cells, that release cytokines and granules. Yeah, we didn't get that. It's important. Mast cells are also important for wound healing and basically for inflammation. When inflammation happens, immunization induces active immunity, the activation of B cells that produce antibodies. So we got that right too. Congrats. We are done with this immune system right here. Perfect. Let's take a break and we'll be back.